I'd like to start by thanking the organizers for the opportunity to present. I'm very excited to tell you about Century's story and our approach to manufacturing, which we feel will be very enabling in terms of making us a leader in this field. First, a little bit about Century. We are a company focused on developing immune cell therapies, particularly NK and T cell to start. These will be derived from IPSCs and genetically modified in a number of ways. We expect, of course, to target the cancer of interest, to have other modifications to modulate the tumor microenvironment, and have modifications to address stealth markers and enable multiple dosing, because our ultimate goal is to provide cures against many types of cancer. Like any allogeneic product, we expect this product to be on demand and off the shelf, readily available. And of course, being able to make large batches of these products in a universal way. We expect these products to be affordable and accessible to many patients. So Century was founded in 2019 with a key deal with Fujifilm Cellular Dynamics, FCDI, which brings key IP to our approach. Most importantly, reprogramming technology built on the IP estate from Jamie Thompson, University of Wisconsin, which allows us to reprogram cells to iPSCs. FCDI also brings technologies to differentiate these cells to key immune effector cells, including T cells and NK cells. And FCDI has a GMP manufacturing facility, which we will utilize for our first products. We were launched with $250 million of funding, combination from Versant Ventures, the Leaps division of Bayer, and Fujifilm. This funding will allow us to build a vertically integrated company, and we've begun to assemble critical mass. We are currently over 90 employees. Our headquarters is in Philadelphia, and we have sites in Seattle and Canada. So I want to start by positioning what we do compared to some other types of approaches. The simplest one to compare is allogeneic versus autologous. And as you probably understand, autologous means every patient has to have his or her own cells processed for them and sent back to them. So when you want to make more for more patients, you have to make more batches. You can't make larger batches. You have to scale out, basically, and make more to satisfy more patients. Not very efficient. With an allogeneic product, meaning that it's a universal product that can be given to any patient, one batch can serve many patients, and we can scale up as much as the cells allow. These economies of scale will give us many advantages. Of course, the raw materials, the labor, the testing costs get spread out over more batches, and this ultimately lowers the cost of goods, means less labor demand, which becomes challenging, especially as you get to commercial scales, and a greater capacity to make more doses to satisfy more patients. Now, if we just contrast the iPSC-derived allogeneic approach to the donor-derived, there's one key difference that leads to a number of important advantages for iPSC-derived. And that key difference is that with iPSC-derived, you have virtually unlimited potential of expansion of those iPSC cells. With donor-derived, being that you're starting with a primary cell, a T cell or an NK cell, there's a limit of how much you can expand those cells. Because of this, there's only so many gene edits you can do on a donor-derived cell before you run out of the expansion potential. With these iPSC-derived, as I mentioned earlier, we have intentions to make multiple gene edits, both knock-ins and knockouts, to get the cell of interest. When you go to scale this up with the donor-derived, you're going to be limited by how much expansion potential you have. So you'll have some scale-up, you'll have a fair amount of doses per batch, lower cost of goods than autologous, but with iPSCs, you can scale up as much as the technology will allow you, which will give you the greatest possible scale-up and the lowest possible cost of goods. And then lastly, because iPSCs can have unlimited potential, we believe we can make a lifetime worth of product from one donor. Therefore, there's no donor-to-donor comparability challenge, and all the gene edits we do, we'll do once up front and be done, as compared to donor-derived, where you'll have to change donors and show comparability and do the gene edits each time you make a donor. So when we think about the one donor for the lifetime of the product concept, that means one master cell bank. So one of the first things I want to talk about is our analytical strategy. And when we think about master cell bank and where it fits, I'll just explain that very simply. This is a rough schematic of what our process looks like. We start with the donor, reprogram cells of any type from that donor to iPSCs and make an iPSC bank. From there, we do our gene edits. And once we are satisfied with our gene edits and all our knock-ins and knockouts, we can select for a clone. From that clone, we still have great expansion potential. We can then make a master cell bank. That master cell bank signifies the start of manufacturing. And from that master cell bank, we can make working banks, et cetera. This schematic depicts making hematopoietic stem cell line from the MCB and then making your NK product from that. But the key here is, being that it's only one master cell bank for the lifetime, our analytical strategy is to very, very comprehensively characterize that master cell bank, especially from a safety point of view. And on this slide, it just gives you a flavor of where the master cell bank testing fits into the bigger picture. Of course, we'll test intermediates, we'll test final products, we'll test donor and some things upstream of the master cell bank. But that master cell bank is so critical because it's the beginning of manufacturing and it has to be safe. So these other tests, and I won't go through them all on the slide, are important, but the safety is the key here, and we will extensively test it to ensure that we do not have the advantageous agents that can bring risk to this product. So now just shifting gears a bit and talking about what are our key enablers. We believe Century will develop two major key core competencies. One is in the cell engineering, which I briefly spoke about, but today's talk I'll focus on the manufacturing competency, making sure we understand how to optimize our processes to get the best possible cells that are consistent from batch to batch, that have the right functional level, and of course good yields to have good cost of goods. And these two competencies, the cell engineering and manufacturing, are interdependent and will feed back to each other to allow us to more expeditiously develop products through clinical trials, and also feed back information for future products that we will make as we learn more about those products in clinical trials. So let's talk a bit about the manufacturing competency now as we go forward. So one major area in terms of making sure that we understand how to make our cells in a high quality way is really understanding our processes. And we plan to have a major investment in process characterization. What this means is, for any given unit operation, we really want to understand how the inputs that we can control affect the outputs, the cell quality. And as we understand this is going to do many things for us. It will allow us to implement processes more successfully, scale them up. As we make process changes, it allows us to do that because we understand how inputs affect outputs, ultimately for better product quality. And all this can be considered quality by design, which ultimately helps with the goal you'll need at commercialization, which is process validation. We'll do the same thing for assays as well, as far as characterizing our assays deeply, making sure we're measuring the right things in the right way. So this will be one major area of focus for us to ensure success in manufacturing. The second area is an investment in building in-house manufacturing. We believe it's so important for a complicated technology like cell therapy, especially these kinds of cell therapies, to have in-house manufacturing. As you make product for clinical trials, there's so much know-how that's gained, and having that in-house allows you to gain that more directly and more comprehensively. So that knowledge we gain is going to be so important for developing that product and future products. And then having that ability to do the manufacturing yourself, we believe will give better manufacturing success, because we're going to have our development and operational teams working together to make process changes, scale up, et cetera, fix problems, optimize, and of course, control the schedule is always a benefit of having your own facility. And we expect to have two. I mentioned earlier, we have access to FCDI's GMP facility in Madison, Wisconsin, and we are also building our own facility in New Jersey with the expectation of it being ready for the first tech transfer by the end of 2021. And it's not just the operations that are important here, it's that collaboration between development groups like process and analytical development and the operational groups, where together, as we implement processes and develop them through clinical trials, we expect to develop a lot of know-how. And then within this diagram, you can see some of the areas of know-how we expect to develop. And I won't get into those details today in the interest of time, but as we generate this kind of know-how, it will greatly benefit our programs and speed our approach through clinical trials, and also inform us as we develop further products. And then the third major area of focus I want to talk about is a focus on scale-up. As I mentioned earlier, allogeneic can be scaled up quite a bit, and iPSC derived can be scaled as much as the cells will allow. But with scale up comes challenges. So if we want to do this successfully, we have to do many things. And here you can see some of the different areas that have to be scaled up. And I'll give some examples on the next few slides of each of these. But no matter what you do, you need to have a major investment in engineering. You have to understand how the physical and chemical parameters of a system affect your cells and control them properly to scale things up. Process characterization I talked about. Product understanding is extremely important. You need to know what you're trying to shoot for, and you need assays that are meaningful and reliable. So let's go through a couple of examples briefly. First is cell expansion. No matter what platform you choose, and here's just some examples of platforms you might choose, there's many others as well, there's always going to be some key scale-up challenges to growing cells. I'll mention them briefly here, and we'll get into the technical details today, but just let you know that these are the things that we are paying attention to, and we are going to put a major focus on to ensure success through scale-up. So getting enough oxygen to our cells, removing CO2, which can be damaging, paying attention to how the dynamic effects of these systems can impart shear damage to our cells, having proper mixing for uniform cultures, and then controlling chemical and physical parameters such as pH properly. These are all very, very challenging scale-up challenges that we have to pay attention to. So the last example I want to cover today is the supply chain challenge for cryopreserved products. These products will be shipped frozen. They'll be taken out of a long-term shipper and put into a dry shipper, which allows you to store the product in the vapor phase of liquid nitrogen, shipped to wherever need to be shipped, and then thawed in a infusion center, administered to the patient. So we'll of course generate stability data to show that cell viability and function is maintained through the freeze and the thaw. And that's a challenge in itself, but I want to talk about the post-thaw challenge. Once the cells are thawed in the clinic, there's going to be some handling to be done, whether it be a dilution or movement from a vial to an infusion bag. Something has to be done, and we'll have to pay attention to post-thaw stability. We'll have to train the sites to ensure that they handle the product and thaw the product the way we expect them to. We believe that if this process is simple, there won't be further release of product needed, further testing needed. We don't want that. We don't want to bring manufacturing to the clinical sites. We want manufacturing to end in our manufacturing site. And we believe that that can be managed with a simple thawing handling process. But the other consideration to think about is the fact that shipping these products is quite expensive. Shipping a dry shipper like this is expensive, both getting it out to the patient, but also getting the dry shipper back to your manufacturing facility or to your provider, because this is a reusable system. That's going to significantly add to the cost of goods per dose. So we'll think about shipping multiple doses if we can. Maybe amenable in clinical trials, may not be, but certainly commercially it's something we want to think about. And lots of questions there. If you were to ship multiple doses to a commercial site, how would you charge the site for those doses? Do you charge them when you ship them? Do you charge them when you use them? What about storage of those samples? Most infusion centers are not equipped for cryopreservation handling. So you can buy them, cryo tanks perhaps, but then you'd have to maintain them. Many, many things to work out still for supply chain. As we think about this, we wonder, would a dry ice -80 supply chain give advantages? Could it be done? Would the cells be able to be stored at -80 and shipped in dry ice? Certainly it would reduce your shipping costs because dry ice containers would not need to be shipped back. But this is something that we're very interested in looking at down the road as well. So just to wrap up, I believe I've told you how Century is building a end-to-end vertically integrated company. We believe we are going to be leaders in the allogeneic cell therapy space based on IPSC derived cells. And 3 main areas of focus in manufacturing, which we really feel will help differentiate us in terms of making high quality, reliable products with speed to clinic and a line of sight to commercialization. The first is a deep process understanding and assay understanding to allow us to have strong products and allow us to make changes along the way. This investment in in-house manufacturing, which we feel is so critical with such a complex technology. And then lastly, having expertise on scale up for all of these different types of operations, which will allow us to produce high levels of product at a reasonable price and make them accessible for many, many patients. Thanks for your attention.